All right, I think I am live. Welcome, ladies and gentlemen, to fall 2024, week 4, BIO201. This is your professor, Dr. Sean Clavel, and I'm here to go over a little bit of a review for week 4, my online and in person students. And so, as you can imagine here, the big topic or associated with week 4 is going to be the exam number 2 over chapters 2 and 3. I'm getting your canvas pulled up here. It is there. It will open up tomorrow. And I'll just give you the you will be hopefully you'll be able to, when you get there, you'll be able to find it there under modules. It is okay. So we can get into a couple of the components here. We're looking at I'm not going to do great math here. 67 times 1.5.
It's 100.5.
Yeah, so it is. It's 100 points. And so the exam here will be, see if I can't pop it up there for you. We're going to be looking at chapter 2, not chapter, well, yeah, chapter 2 and chapter 3 a test. It's exam number 2. I'll fix that right quick here. And so here we go. Let's see if I can't break that. There. I don't know why that didn't work there. Let me refresh this slide. There it is. Exam 2 and 3. It will have 67 questions. So I think our previous one had 50 questions. And it was over chapter 1. Chapter exam number 2 over chapters 2 and 3 will have 67 questions. It'll be about a point and a half each, and so if you multiply that out, it comes out to 105.5 points total. So there will be multiple choice, there will be multiple answer, there'll be some very short answer as well. Again, I won't ask you to write more than a brief description, so 1 or 2 word answers are my short answers there, and you're hopefully familiar with some multiple choice and multiple answer type questions there. Let me take a look here as to some of the topics that we should be reviewing as it relates to some of these, as it relates to the chapter 2, starting with the basic chemistry, kind of break it down into the basic chemistry, so knowing some basics about some of the elements, particularly the elements that make up a majority of the body by mass, the structure of those elements. So they're made up of subatomic particles that include electrons, neutrons, and protons. Where are they located? What is their relative mass? And how ultimately does the interaction of electrons allow for chemical bonds to occur? And then chemical reactions are, metabolism is all of the chemical reactions that occur in the body. Millions, if not more, at any given time. And so understanding that it's, homeostasis is a process of ebb and flow where we modulate around a set point. it's not a direct set point. There's usually a high range and a low range. And so all of the chemical reactions ultimately turn out to be either ones that synthesize or build up tissues in the body, or ones that break down. And then the inorganic and organic compounds, thinking about water and its special properties. And then the organic compounds, specifically carbohydrates, proteins, fats, and nucleic acids. Again, this is a chemistry review. It's right there in the book, chapter 2. Again, encourage you to start just reading it to start with, reviewing it in terms of the outline. The best text that I've used so far is the Merriam-Webster. And again, I can send you the resources there, but hopefully you guys are familiar with that by now. Chapter 3 is when we take a look here at cell biology. So some of that 181 material that we review here. So chapter 3 starts, it's a basic overview of cell biology. So thinking about things from cell theory, right? All living things are composed of cells. They arise from pre-existing cells, mitosis. Though we won't be talking about meiosis here. But mitosis specifically, what are the stages of mitosis? What is the stages of a cell life and cell cycle? What are the parts of a cell? And so we'll jump into the A. We'll be looking at these. I gotta fix that slide, huh? Different tissues in the body in terms of structure and function. So we'll be looking at the kind of the physiology of skin and physiology of bone, and we'll be also looking at its structure as well. What are the components that we call it in terms of its structure? So when we talk about the structure of the cell, there's the plasma membrane. Its function is to act as a barrier and regulate movement of substances. The cytoplasm is where we have the organelles. It's divided into the fluid component, that is the cytosol, and the organelles, right? The material that is suspended within this fluid. Staying on the topic of the plasma membrane, we talked a little bit about what's known as membrane dynamics, or the activity that occurs at the membrane. Plasma membrane, cell membrane, it's all the same thing there. It's kind of fluid mosaic. When I mean fluid mosaic, it's kind of like if I were to put like a pin through that cell, the cell membrane would just encompass around it, right? And then I can pull it back out, and that would close back down. So it's a fluid mosaic. So it can kind of be split a little bit, but also it'll adhere back into its shape if I were to put like a very fine needle into it. But there is some activities that occur within the cell membrane, right? And they think about things that include the transport of water, or osmosis, the diffusion of water and other chemicals across the cell membrane. That's traditionally diffusion and osmosis occur through from a level of high concentration to a level of low concentration. So thinking about the idea of the sodium-potassium pump. And then opposite of that, when we go from areas of low concentration to areas of high concentration, that is related to that's going to require energy. So those are going to be our active transport systems. We think about endo and exocytosis when that occurs, but we also think about these membranes, these membrane proteins that require energy and move contents against their gradient. So sodium and potassium get moved across their gradient. The thing that you might want to write down when you think about what is going to occur with the chemicals inside and outside of the cell is this. Let me see if I can't get something to write with. Sorry, guys. Okay. Click that. Click that. Click this. Click this. Okay. Getting something to write with. Here we go. Screen 2. Yeah, we'll pull that away. Okay. All right. So, the thing that I often write here is so let's first talk about the contents, the abbreviations here when we talk about. First is the extracellular fluid, so the outside of the cell, and then the intracellular fluid, so the inside of the cell, right? And so, like as we've mentioned, there is a membrane, plasma membrane that allows semi-permeable membrane that allows things, certain things to pass and others not. And on the inside of the cell, let's say the inside, there is a high concentration of potassium. So okay, put that plus there, and then it's often, I'm gonna put this here, make it so. Put an up arrow here. High concentration of potassium inside of the cell. There is a high concentration of sodium inside of the, high concentration of potassium inside of the cell. High concentration of potassium, why am I saying that? Of sodium outside of the cell. This is right on the screen. And so again, remember that the sodium potassium pump requires energy, and it's responsible for bringing 3 sodium ions into the extracellular space, into the extracellular fluid, and drawing 2 potassium ions into the cell. And so again, because this goes from concentration gradients, right? Even the 3 sodium are getting kicked out, even though there's a high concentration already, and 2 potassium are getting brought in, even though there's a high concentration already. This does require energy, so it's going to require ATP to energize this endeavor. So the sodium potassium pump is responsible for, again, maintaining the concentration inside and outside. And now that you have a chemical gradient, there's also what's known as a electrical gradient. So we'll see here in what's maintained in irritable or excitable cells is a concentration gradient on the inside, or an electrical gradient on the inside of the cell, somewhere in the range of negative 70 millivolts. And what that does is that allows the little charge to stay on the outside of the cell, so when your nerve sends a signal, it just propagates, as the term, an action potential propagates across the cell membrane. And so that charge is what allows that signal to move at the speed of electricity. And so, yeah, being familiar with the sodium potassium pump is going to be a big one. Let's stay here. Let's continue on. Membranous organelles and non-membranous organelles. Let me jump back here to one more thing here. because I want to mention this too, is the topics related to osmosis, right? What happens with water. So let me make it a different color. Let's make it a highlight style. This topic here, osmosis. So what we're talking about is where solute goes, water is going to follow. So if there's more solute inside of the cell, particularly like red blood cells that are suspended in an aqueous solution, what I mean by an aqueous solution, water. So red blood cells suspended in an aqueous solution may have to have a particular concentration of solute inside and outside of the cell, because water can then freely move in and out of the cell. It doesn't have to be the same particular solutes, but the same amount or same concentration. And so what happens if someone is infused with distilled water or free water, it will dilute the aqueous solution around those red blood cells and would cause water to rush into those cells, referred to as hemolysis. So being able to understand what what tonicity occurs. So the term tonicity or tension that is built because of the concentrations of solute, whether it's inside or outside of the cell. And so we usually refer to it as the concentration outside of the cell. So if it is a hypertonic solution, there's more solute outside of the cell. If it is a hypotonic solution, there's less solute outside of the cell. And so being able to identify the associated what do I want to say? The associated things that would occur, whether it's a hypotonic or hypertonic solution. Now, an isotonic is the ideal solution, where there's enough, there's an even distribution of solute inside and outside of the cell, and water can then be pulled in and out and allow for a normal cell shape. Some of the other things that give cells shape include organelles. Clear all my drawings. Include the organelles. And so non-membranous organelles specifically include things like the cytoskeleton. Those are protein structures that give the cell structure. So not too many cells in the body other than sperm cells have flagella, but cilia are some hair-like structures that we'll find in epithelial cells that move substances one way or up, one way or another down a passageway. So cilia are some of those structures, but also proteins embedded into the membrane would give it structure as well. But these non-membranous organelles are structures that do not maintain a traditional membrane, like we would think like the endoplasmic reticulum or mitochondria and lysosomes. These are some of those structures that have a distinct shape or feature to them. The non-membranous organelles include, again, the cytoskeleton structures and ribosomes particularly. There's two more slides here. The chapter 3 also talked about the nucleus and genetic information. So the control center of the cell is the nucleus. Why? Because it houses DNA. What is DNA? DNA is the original information. It's the original code for creating all of the proteins in the body, and ultimately all of the whatever distinct proteins that are created within a specific cell is what that cell is going to become. Muscle cells have very distinct fibers, right? They're malleable type of protein fibers, and so what that happens is if you stress them or exercise them, they will ultimately rebuild those protein fibers a little bit bigger and stronger. Other cells, like your skin cells, will revitalize and replenish, but after a few weeks they'll die off. So they go through a very short cell cycle, while things like your nerves have a very long cycle, right? And so the nucleus of the nerves are created or provide for a genetic code that allow for healing of the myelin sheaths, as well as other things that would maintain a cell for a lifetime. And then finally, the cell life cycle, right? So, thinking about breaking down the cell life cycle into two phases. First is interphase. Interphase is G1, the synthesis phase in G2. So, particularly, we want to highlight here that, I'll make it a different color. This synthesis phase here is where we're going to have DNA replication occur. So, DNA replicates prior to mitosis, which is the splitting of or the cell division. And so G1 is a preparatory phase for the replication of DNA, and then G2 is a preparatory phase for the preparation of mitosis. And then ultimately, the 4 stages of mitosis: prophase, metaphase, anaphase, and telophase. And then a little bit here on some cancer and cell division, which we'll talk about it again in chapters 4 and 5. Yeah, you know what? I'd be remiss if I didn't give you guys a couple of practice questions here. So, let me finish this off here with a little bit of love for all the ones that stayed here and watched it all at the end. Here we go. Let's draw this back. What's going on here? Excuse me. Okay, so let me get into the exam number 2. Alright. Alright, so here we go. Let's give you about 4 or 5 questions here. Here we go. So the movement of molecules from high concentration to an area of low concentration. There are 5. There's 5 possible answers here associated with that. From high concentration to low concentration, right? Not going to give you the answer though. The movement of molecules from a high concentration area to a low concentration area. Which one of those is associated with that? The rough endoplasmic reticulum, named due to what associated structure primarily? Why is it? What makes it rough, I guess? A substance dissociates into a potassium ion and the chloride ion in a solution. This substance is a blank. You've got 4 options there. A substance that dissociates into potassium ion and the chloride ion in a solution. The substance is a what? Which of the following best describes the electrolytes that are dissolved in the body? Is it 1, 2, or 4? And I don't know if I like that one. The bonding of calcium, phosphorus, and other elements produces mineral crystals that are in bone. This is an example of a blank reaction. Bonding of calcium, phosphorus, and other elements produces mineral crystals that are in bone of a blank reaction. I think that's 5. That is 5. Let's wrap that up there. How do I get out of this preview? I'll just pull down the screen. All right. There you have it. Ladies and gentlemen, I'll wrap up my announcements here. I wish all y'all the best of the rest of the week here as we wrap up week number 4, particularly exam number 2, chapters 2 and 3. You got the amount of questions there. Oh, I think you got 75 minutes, 80 minutes to complete. And I wish y'all all the best. I appreciate all of your input and your work so far this semester, and I look forward to all of your responses now or coming up and all throughout the semester. Thank you guys, and have a good evening.